All right, I'd like to introduce John Adams to you. He's spent 2 years me torturing him. He's going to do his thesis presentation on the effects of prescribed fire on tick-borne disease ecology and small mammal ecology.
All right, thanks, Dr. Carter. I'd like to thank everybody for coming that's here. I'm going to try my best to cover this pretty quick. There's a lot of information. We had a lot of stuff we covered. I don't want to keep y'all in here for over an hour, because if I just started talking, you'll probably be here for a lot longer than that. Small mammal communities, they comprise an important part of most of the vertebrate biomass and biodiversity of forests. And they influence the vegetation structure through consumption and dispersal of seeds and fungi. They're also an important food source for many predators of the forest. And many mammals associated with early succession depend on disturbances to create the suitable habitat. Some of these disturbances can be created naturally by fire or anthropogenically by prescribed fire. Prescribed burning treatments result in changes in important components of small mammal habitat. It can lead to increased herbaceous vegetation, decreased shrub density, and decreased woody debris. So overall, forest thinning by prescribed fire or mechanical means can be expected to increase densities of small mammals, and retention of slash and fuel reduction restoration treatments may further increase small mammal densities in a post-treatment community. Allgreen in Minnesota and Sullivan and Botain in British Columbia saw dramatic increases in Peromyscus species on burn sites following fires. This is presumably because of the rodents' ability to forage for seeds and insects was greatly increased. Higher Peromyscus species populations on burn sites have also been attributed to better visibility and abundance of seed, food source for the mice, after reductions in litter cover and depth. Basically what it does when you have a fire, it burns away a lot of your ground cover, the seeds are visible. Also, it's been shown that the ground cover, it will be unburned patches in your patches of burned area, and that's like a buffet for them right there. Small mammal populations have been shown to be affected by the amount of coarse woody debris, or CWD, in the area. I may refer to it either way. I'm not going to say which way. CWD is any standing dead tree, down trunk, or fallen or down large branch greater than 10 centimeters in diameter. These fallen logs appear to be selected as travel routes, orientation aids, cover, foraging, and nest sites. The primary host species of tick species is mammals. To serve as an efficient reservoir, a host species must be locally abundant, have many locally infected individuals that serve as hosts, and not become resistant to repeated tick feedings. Hosts also have to be easily infected, maintain the infection for long periods of time, possibly life, and have limited home ranges that result in continued residence in tick-infested areas. The basis of this. Lyme's disease is the most commonly reported vector-borne disease in the United States. It's caused by Borrelia burgdorferi. The other disease is a clinical condition similar to Lyme disease, referred to as southern tick-associated rash illness. It's associated with the bite of the Lone Star tick, and they carry the causative agent of the disease, Borrelia lone stari. In the coastal northeastern United States, the primary wildlife reservoir for Borrelia burgdorferi is the Peromyscus leucopus, which is the white-footed mouse. In the eastern and central United States, the white-footed mouse is also usually the most competent reservoir for Borrelia burgdorferi. But also, the chipmunk can also serve as a significant reservoir for burgdorferi, particularly during times when mouse populations are reduced. Also, in a study by Moore et al. 2002, deer are a major host of the Lone Star tick, and they also had sequence confirmed evidence of Lone Star flagellin gene by PCR, indicating that deer are infected with Borrelia Lone Star Eye. It has not been proven that small mammals are the major host of the Lone Star tick, so therefore, the likelihood of them being vectors for Borrelia Lone Star Eye needs to be researched further. This study investigates the effects of prescribed burning on the ecology of small mammal communities. It also will examine for infection of Lone Star Eye in mammals within this prescribed burning ecosystem. And it is hypothesized that mammal communities will increase in diversity and richness immediately after the fire, followed by a decrease in species richness and diversity as time from the fire increases. So, the study area. It's Talladega National Forest, Shoal Creek District. It's located near Coleman Lake, which most of y'all, I'm pretty sure, know where that is. And it's comprised of upland hills with low mountains that have largely steep slopes. Prescribed burning is conducted routinely in this for the management of longleaf pine, northern bobwhite, and red cockaded woodpeckers. So we chose, my study area had 12 plots. They were chosen based on length of time elapsed since prescribed burn. I had 3 plots of each treatment type, the 4 treatment types, I'm sorry. It was 1 month post-burn, 2 years post-burn, 5 years post-burn, and then I had an unburned control that was burned in excess of 15 plus years. I established 40 by 40 meter plots in each treatment type. What you're looking at here is just some examples of the sites. The upper left-hand corner is the 1 month since burn treatment. The one below that is 2 years since treatment. The upper right is 5 years since treatment, and 15 years for the bottom right. If you'll notice, you can see the big increase, the change. You can actually see the change from the first slide to the end of the decrease amount of ground vegetation, and just the general in the newly burned sites, the amount of ground vegetation that's there, just lots of it. All right, for the mammal trapping, a 3 by 3 trap grid with 20 meter spacing was established in each of the plots with a total of 3 grids per treatment type. Trapping sessions were conducted for 5 nights out of every month for a year, starting in April 2008 and continuing to April 2009. Traps were set and checked in the morning. Trap sessions were also conducted based on moon phase, typically near the new to quarter moon. I did this due to most of the mouse are nocturnal feeders, so you want to not do it on the really bright, like full moon nights, just for increased predation risk on the mice. They don't seem to be as active. The mammals were captured, were identified the species, ear tagged, and a tissue sample was collected from a tail clip. Animals were also combed for ticks. And I don't know if anybody else has ever done that, but that was really a tedious process. Those little guys like to bite you. They don't want to be friendly. Only animals that were newly captured or were not recaptured in the same week were examined for ticks. So if I caught the same mouse in the same week, I didn't recheck it for ticks. Ticks and tail samples collected were examined for the spirochetes, that are the causative agents of star eye, which is Borrelia Lone Star Eye. That's just a picture of a Paramiscus right there in the picture. So we can see the ear tag in his ear, little silver piece. Also did a microhabitat assessment. A survey of vegetation was conducted by using a modified Carolina vegetation survey. Canopy cover was measured using a spherical densitometer, and soil moisture was obtained using a soil moisture probe. Coarse woody debris. So you had 3 non-overlapping transects. Coarse woody debris greater than 10 centimeters at point of contact with the line was measured. Length and diameter were measured. The diameter is the diameter at breast height, which on the ground is 1.4 meters. And the volume of the coarse woody debris was estimated using the diameter at breast height model. I'm not going to explain the formula to you. You see it there. It's a lot of LN. For statistical analysis, the Shannon-Weiner Index of Diversity was used to measure species diversity. Species composition was also analyzed using Mann-Whitney test and a diversity t-test to determine statistical significance between treatments.